Hello everyone, back to my channel, and today I'm going to discuss about the structure of chromosome. Basically in this video I'm going to discuss about a brief description about the structure of chromosome. I will do the structural organization of the chromosome in a later on video, but let's dive into the topic. Let's get started. So each of the chromosome having two identical chromatids which are connected through the centromere, which is signed as a blue dot over here. Basically what is happening, that two chromatids are connected via centromere, which forms a chromosome. Now let's talk about what are the chromatids. Now, as I earlier said, that the chromatids are one of the two halves of the chromosome. They are identical halves. Basically, from if two of the chromatids are generated from one chromosome, then they are called sister chromatids. And the centromere of each chromatid divides the chromatids into two arms, basically the shorter arm and the longer arm. I will discuss this thing in a later on video where I will discuss about the classification of the chromosome on the basis of the position of centromere inside the chromosome. Now, the shorter arm is named as a P arm and the longer arm named as a Q arm. So basically what happens, at the time of cell division on a prometaphase or the metaphase phase, when the chromosomes are coiled up and they are highly coiled and they are dense, so we can see this structure on the microscope. Under the microscope, if we strain the chromosome, then so these are the chromatids which you are having, and this is the centromere. Now, the centromere of the chromosome is also called the primary construction of the chromosome. Basically, the centromere is also associated with the mitotic spindle attachment on the chromosome. This on the centromere, there is a protein made disc-shaped part which is called kinetochore. Basically, the kinetochore is the point where the mitotic spindles attach in the time of cell division, and they helps in pulling the chromatids to the opposite poles. Here I have shown this picture. This chromatid is pulled in this side and this chromatid is pulled in this side. So basically they are pulled in the opposite direction to the poles of the cell division. As I have earlier said that chromosome having two identical halves, so let's talk about the single half of a chromosome. So that is a chromatid. So chromatid is having different parts, just like primary construction and centromere, which I was discussing in the earlier. The chromonemata, which is a thread inside the chromosome chromatid. The chromomeres, which is a bead on string structure. Then the telomere of the chromosome, the secondary construction of the chromosome. A chromosome may be having one or two secondary constructions. One of the constructions is also known as the nuclear organizer region. Basically, this region is only responsible for the generation of nucleolus inside the nucleus. And here is the satellite region. Basically, satellite region of a chromatid is a kind of it. Sometimes it may be there, sometimes it may not be there. Basically, for a human genome, the 13, 14, 15, 21st, 22nd, and the Y chromosome is having the satellite. Rest of the chromosomes doesn't having the satellite. Satellite chromosomes are also called SAT. SAT stands for Sine Acid Thymonucleonico. I will discuss it on a later video. So move towards the next. Now the centromere. Centromere is the special position on the chromatid which contains special DNA sequence of chromosome that links a pair of sister chromatids. Basically, it helps the cohesion complex which mediates sister chromatid cohesion after the DNA replication, as well as coordinating the sister chromatids separation during the anaphase. Now, what is kinetochore? Kinetochore is a disc-shaped protein structure associated with the duplicated chromatids. Kinetochore, at the time of cell division, the spindle fiber attaches and it pulls the sister chromatids opposite direction towards the poles. The centromere is also called the primary construction of the chromatid. Now, moving towards the secondary construction of chromatids. It is the another construction on the chromatid which contains gene for construction of nucleoli. Basically, I have earlier said that it is also called the nucleolar organizer region or NOR. It can only be identified in the anaphase because only the bending will be at the centromere. So, if there is any another construction except the primary construction, then that is the secondary construction or the nuclear organizer region. Next, we have the satellite and the telomere. So, satellite is the elongated segment. It is also the present in secondary construction. Not every chromosome has satellite. I have earlier told this thing. And the telomere is the terminal part of the chromatid. The telomeres are polar segment which prevents the fusion of chromosome segments. Every chromosome has the telomere, but every chromosome doesn't have a satellite. So this is the key difference between satellite and telomere, both present in the terminal end of the chromosome, but telomere is present in every chromosome, but the satellites are not present in every chromosome. Now, the chromomere and the chromonemata. So, what is chromonemata at first? At the prophase, the chromosomal materials look like a thread structure. Basically, I have shown here a diagram and I zoomed it and it looks like this. So, this is the thread-like structure that is called the chromonemata. It's composed of DNA, RNA, protein, and many things. And the beads, the small loops on it are called chromomeres. At interface, it is called the bead-like structures. Now, what happens during the cell division? The chromatins are get condensed. So you know that inside the nucleus there is a chromatin fibers. The chromatin gets condensed, and if we strain it, we can see there are two regions we can be highlighting under the microscope. One, a portion of chromatin, it is darkly strained, and a portion of chromatin, it's lightly strained. So the darkly strained area, it is called the heterochromatin, which is genetically inactive, and the lightly strained area is called the euchromatin, which is genetically active. So at interphase, the chromatins coils up, and then they are slowly forms the chromosome, and the chromosomes are getting visible at the metaphase stage. But during the interphase, we can see the chromomeres and the chromonemata. So in the interphase, when we can see the chromonematas and chromomeres upon it, so it looks like necklace with beads or beads on a string structure. So what happens is when the interface comes up, the chromomeres are visible and they are highly coiled. But at the time when it goes for the prophase and they are forming the chromosomes, then the chromonematas, the thread-like structures, and then they are going to visible. So that happens. I hope you guys enjoyed this little video on the structure of chromosome. On a next video, I will discuss about the structural organization of the chromosome. Till then, stay tuned.